The point where those nerve fibers converge and we get the formation of the optic nerve. Basically what's going to happen, we're going to see the light ray enter the eye. It'll focus on the retina. When it does that, it produces, yes, an inverted image, upside down, okay? The pupils, they have to respond to the light coming in because it is under the control of the autonomic nervous system. What we end up getting for the control of the pupils and the light coming in is something called the photopupillary reflex. For example, if you cover one eye, which would be, in no light, you would think that pupil would be what? Constricted or dilated? It's going to be dilated because this one is. If it's in less light, it should constrict. But because they respond the same, whether one eye is covered or not, both pupils, will always be the same. Well, depending on certain circumstances, the pupils should be the same on each eye. Okay? Certain medications, drugs, that sort of thing can affect this particular reflex. That formation of the image does depend on all parts of that eye bending or refracting the light. The cornea is going to have a major input on refracting the light, and the lens will help to fine-tune that image. When we look at any type of defects in an image, those can require the cases where we may need glasses, contact lens. Some people go so far as to have like the laser surgery and so forth. That's when when the lens of the eye doesn't quite do what we want it to do. And we end up with stuff such as astigmatism, nearsighted, farsighted, that sort of thing. Most of the time, glasses or contact lens, their job is to change the way the light would come in and refract, change that so that it hits on the retina correctly for you to be able to have the correct image. Okay? Making your vision clear, better. The cells that are going to be responsible for the action potentials that are going to pass along the optic nerve, these are our photoreceptor cells because it is the fact that they have light energy coming to them. And they have to take that energy and create the electrical and or chemical signal that the nerve fibers can understand. Rods and cones are the ones, now we're going to see other cells involved, but rods and cones are the cells that give us images. They give us vision. Rods are for night vision. Is your vision really clear at nighttime? You're walking through the house and, that sort of thing, you bump into stuff. It's because the image is not as clear. Cones are the ones that give us the clearer image. So guess where they are highly numbered in location?
Fovea.
The fovea centralis, the pit. The pit has a very high number of cones. They're termed rod and cones because of the way that they're shaped. Now, they do have a peculiar shape, okay, altogether. We're going to find, however, that there are other cells that are going to be involved. But just like we've studied up to this point, we always have the nerve cell that's going to send the signal, okay? The rest are supporting cells, okay? So rods and cones are going to be cells involved in the makeup of the eye that are going to be sending or giving us the image. Other cells that are involved are termed bipolar, ganglion, horizontal, and amacrine. They are supporting cells. However, they're important in the passing of the information to form the image. Here's pretty much what's going to happen for us to form an image. Note how they're trying to show us the back of the eye, the light coming in. The light coming in, well, we're going to see the light move through the parts of the eye, and we're going to begin to make the way to my cells important for vision, my rods and my cones, okay? The rods and the cones, well, pigment epithelium, choroid, okay, highly pigmented, all right? So here's, look at how it's going. Does it look like it's going back or front? For the light coming in. Kind of looks like it works in reverse maybe. Okay? Sort of, but you got to remember the tunics that are present. Okay? Now, in the retina, the neural component. All right? Well, if you remember, behind it, I've got my pigmented epithelium. Embedded into that are my rods and my cones. So light gets to come in, rods and cones pick up whether we're dealing with a huge amount of light or little, so for example at nighttime. They are going to begin to have synapsing onto these other cells. These other cells are not responsible for the vision. They are simply support cells, mainly because the eye cells are very busy, okay? So you can see that they will synapse onto a bipolar cell and a horizontal cell and the amacrine cell. Now, bipolar, this is one of the two areas that a bipolar cell is found in the body. The other area is the sense of smell. Okay? Now, as we begin to have the stimulus get picked up, they're trying to show me that I synapse onto these other cells the direction that the stimulus is going to go. So my stimulus continues to move down. As I move down, like through the bipolar cells, I'm going to end up getting to a ganglion. Do you happen to remember what a ganglion is?
Nerve cell body.
Where we've got some nerve cell bodies. So now, these supporting cells, they get to synapse onto dendrites into the area of the ganglion. The ganglion, which is receiving input from the multiple support cells, okay, that ganglion, the fibers begin to exit, go down the optic nerve to the area of the brain and that occipital lobe to make the image. And we see.
So the light essentially passes by all of the directional information
All of the support cells.
and then is returned to it to present us with an image.
Yes, that's a good
That's
way
what
to
you. That's what you meant by working reverse.
It looks like
When you
it's working backwards.
I wasn't sure what you meant when you asked that. I was like,
Now, your textbook is going to get very detailed about how the rods and the cones and these chemicals like the rhodopsin, iodopsin, and so forth are going to wash and create the bleaching of. I'm not asking you to know that. What I have just explained is what I want you to know. I'm not asking you about the rhodopsin. I'm not asking you about the process that's occurring. I want you to understand the cells involved and how we form that image. Okay? Any questions about 16? All right, we're done. 16.
Woohoo!